Okay, so very good morning, good afternoon, and good evening, folks joining from across the world, as we can see. Myself, Urmi Bajpayee from IBRC, and Jessica Secho from Phage Directory, and Yon is also here with us. So we welcome you both. We both welcome you to this second episode of Bacteriophage Bioinformatics webinar series. And as you know that we started this series because we know this fact that there is high throughput sequencing technologies which have come up, and the costs of sequencing has gone down drastically, which has enabled bacteriophage genome sequencing at a rather high pace, like genomes from the other organisms. However, we still don't know very well what a large part of phage genomes do and what do they code for. So acknowledging this importance of genomic characterization while understanding phages, we are co-hosting this series where we are trying to invite guest speakers who are experts in understanding phage genomes. And as you know, you would appreciate, most of us, those who are doing phage research at any stage, we do appreciate that gene prediction and annotation tools which we use, these are customized for phage genomes. Since the available tools, whatever we use them, they are adapted to run on phage genomes, but perhaps are unable to capture their full complexity. So to address this gap, today we have a very special guest. We are very happy to have Ms. Katelyn McNair. She has developed and released a software called PHANOTATE, as you can see on her title. And she's the first gene finder. This PHANOTATE is the first gene finder which is specifically designed for phage genome annotation. And also of interest to phage researchers is her previous tool called PHAX, which computationally predicts whether a phage is temperate or virulent. So Katelyn is doing PhD in a joint doctoral program between the University of California and San Diego University, USA. Her work mostly focuses on big data and applying machine learning techniques to analyze and predict various aspects of phages, as we can see. So perhaps she's planned to go over how to properly manually curate a phage genome, a small genome she has brought to you, via its intrinsic genetic properties. And this would then lead her to introduce PHANOTATE, the software which she has developed and released, which is fantastic indeed. And this PHANOTATE, of course, take these genetic properties, and then she'll try to teach you how to computationally curate a phage genome. So with that, please join me and Jessica and Yon in welcoming her, and she's going to take us through genome to gene to function. So I welcome you, Katelyn, and you may start. Thank you very much for accepting our invitation.
Hello, everyone. And so is my PHANOTATE, the gene finding phages, showing the right screen? Is that on the screen PowerPoint?
Yeah, we can see this.
Okay, cool. So thank you for coming to my webinar, and hopefully you'll find it informative. And by the end, you'll be able to annotate your own phage genome. So a little bit of background. I'm going to be covering some molecular biology, very introductory, and that's of phage genomes, and techniques of molecular biology and how they relate to PHANOTATE. This will be important for both manual annotation as well as verifying automated annotations. So verifying automated annotations is important because programs aren't 100% correct always. Certainly not my program just yet, but I'm trying to get it there. And so usually you have to manually annotate after you've automated to go back and remove any false positives or add any false negatives. In the next part, and then I'm going to go over manual genome annotation using Artemis. And this is a pretty simple to use point and click type software. I'm going to be showing how to annotate the phage genome PHAX174. And then I'm going to go over how to automatically annotate this genome using my software PHANOTATE, as well as the Patrick online repository, which kind of does stuff online and does everything automatically for you. So starting off with the history of gene finding. The first fully sequenced genome was actually a phage Escherichia virus, MS2, in 1976. I released my program PHANOTATE a little over a year ago. So what are all the previous methods and software for gene finding in phages that have been developed in the last 40 years? There are three. Genomark was the first released in the 90s, followed by Glimmer a few years later, and both used previously annotated reference genomes to create gene models to predict genes in a new genome. Prodigal, most recent, is reference-free if your DNA is longer than 10 kilobases, and it looks for consensus gene signal biases. Now you might be thinking, well, what about this other program that I know that does genome annotation? Well, the reality is that most of the other programs out there are really just pipelines that run one of these three programs. This is things like Proca, Genius, Patrick, RAS, etc. So the issue with the previous methods, Genemark, Glimmer, Prodigal, is that they were developed for bacteria, which have quite a different genome layout than phages. The reason for this is that bacteria are essentially big sacks of water with no real restriction on how long their DNA can be. Phages, on the other hand, have a rigid capsid in which they can fit only so much DNA or RNA into. There we go. So shown here are portions of a bacterial and a phage genome. The horizontal line is the DNA backbone of the genome, and the arrows represent genes. The red arrows are genes on the forward strand, and the blue are on the reverse strand. Sorry, my computer's messing up. So the difference between bacteria and phage genome organization is pretty apparent. So bacteria tend to have much longer genes, and their genes also tend to occur in different directions. In contrast, phage genes tend to all run in the same direction. And when a strand switch does occur, all the next subsequent genes are all in the same direction. So another difference is that bacterial genes are generally separated by intergenic regions, while genes in phages have very little spacing between the genes. In fact, genes in phages tend to partially overlap. This complicates gene prediction in phages since genes are not clearly delineated by these intergenic regions. However, what if rather than hindering gene prediction, we use this very strict gene organization of phages to our advantage? This is the motivation behind my tool, PHANOTATE. But first, some background on molecular biology. It's just not.
Are they just not advancing, Katelyn?
Yeah.
Right, okay.
So this is a central dogma of molecular biology, which is DNA to RNA to protein. In the first step, transcription, RNA polymerase transcribes portions of the DNA into RNA. The second step, translation, is when ribosomes find genes in the RNA and translate them into amino acids. So this is an oversimplification because eukaryotes have introns and exons, whereby after RNA is transcribed, it is chopped up and spliced and put together differently to form before it is translated into proteins. This complicates gene finding in eukaryotes because their genes are fragmented across the DNA. In contrast, phages and bacteria do not have introns and exons for the most part. So the sequence of a gene in an RNA transcript is a one-to-one mirror of that sequence in the DNA. Now this collinearity in the DNA allows us to forego the RNA step and look for genes directly in the DNA. This shortcut is why in my slides, I refer to codons as having thymines, when in reality the T in DNA is replaced by U, uracil, and RNA. So when I say ATG is a codon, I really mean AUG. This brings me to the basic layout of a protein encoding gene, which is a start codon, ATG in this case, a stretch of codons that encode for amino acids, terminated by a stop codon, TAA. For prokaryotes, the most frequent start codons are ATG, GTG, and TTG, while the stop codons are TAA, TAG, and TGA. though there are exceptions to these, and their usage changes based on the GC content of a genome. So genes occur in what are known as open reading frames, which are simply a stretch of codons between two stop codons. So shown here, two stop codons, TAA, and are designated with an asterisk symbol, which is the symbol used for stop codons. So the stretch between them is an open reading frame, or ORF. So in order to find potential genes, all we need to do is look for a start codon, or look for stop codons and look for start codon between them, and then follow it to the next stop codon. This gives a potential coding gene, sometimes also called the potential coding ORF. Because codons are composed of nucleotide triplets, this means that there are 3 frames to look through to find potential genes. And amino acid completely changes just by shifting 1 base. Additionally, there are 3 more frames in the reverse direction, which I don't really show here. So time to look at some real data. And this is, here I'm using Artemis by Sanger, which I'll be going over later, and it's freeware, so you can download and use it yourself. And I'm showing the first 8 kilobases of phage lambda. So shown are the 6 different frames, 3 forward on top and 3 reverse on the bottom. The vertical black lines in each of the frames are stop codons. The boxes are open reading frames that have a start codon. They're colored green if they code for a gene, and gray if they do not. There's text on the screen, okay. So if they do not, and they're sparsely coding. This means that the box in the forward frames, the left side of the box is a start codon, and the right is a stop codon. For the ORFs in the bottom, the reverse frames, it's the other way around. There's a stop codon on the left and a start codon on the right. So the first thing you will notice is that the green coding ORFs tend to be much longer, but not always. So this tells us that length is important. The next thing you'll notice is that the coding ORFs are all in the same direction, which is also important. And lastly, you might have noticed that for the most part, when one coding ORF ends, another begins. So using this knowledge, I came up with a model to follow an optimal path through the genes of the genome, thereby annotating it effectively. So my inspiration was a traveling salesman problem. So if you haven't heard of this, it's a popular topic in machine learning, and it goes like this. So there's a salesman. He has a list of cities and knows the distances between each pair of cities. He wants to visit each city once, taking the shortest possible route. To find a solution is a very computationally difficult and time-consuming problem, but luckily mine's a little bit different. I don't really want to visit every city. I just want to go from one side to the other in the shortest path possible. So this gives me different options to use for different graph solving algorithms. In my model, there are 3 connection types. ORFs are favorable steps. That's going from a start codon to a stop codon. Gaps and overlaps are unfavorable. They're yellow, so they're bad. And so that's going from a stop codon to a start codon. And in the case of an overlap, it's going backward, and the gap is going forward. So in this one, open reading frames are good, they're green, overlaps are bad, yellow, and gaps are worse and red. Additionally, there's also strand switches, which are really bad. And these are ORF to ORF connections, gaps or overlaps that go from an ORF in one strand to an ORF in the reverse frames. So to get distance measures for the various connections, I rely on the probability of an ORF occurring by chance alone. We know that out of this possible the 64 possible codons, 3 are stop codons. This means that the probability of encountering a stop codon is 3 divided by 64, or 5%. Conversely, the probability of not encountering a stop codon is 1 minus that, so 95%. Therefore, the probability of an ORF occurring by chance alone is this P not stop, exponentiated by the number of codons. The 3 divided by 64 calculation here only applies if the nucleotides ACTG are evenly distributed. This is not always the case. The GC content ratio changes from genome to genome. It can vary from as low as 25% to as high as 75%. This also affects the probability of encountering stop codons at random. If you have a lot of G's and C's in your genome, you'll see the 3 stop codons, which are mainly composed of T's and A's, less frequently. To calculate the p-stop from GC content, each individual probability of seeing that particular stop codon is summed. So the probability of seeing TGA is the probability of seeing a T times the probability of seeing an A times the probability of seeing a, or the probability of seeing a T times the probability of seeing a G times the probability of seeing an A. And likewise for the other two. The P not stop is the same before, as is the probability of seeing an ORF. So for my problem, now I just need to convert the probabilities into distances. Oh, that came out unformatted. Because ORFs are always favorable connections, this means that they should always be negative, which is, so I show the negative here, it got poorly formatted. I also take the multiplicative inverse, which is essentially dividing 1 by that P not stop, so that the length of the ORF goes up as a distance weight, so that as length of the ORF goes up as the P goes down. So I still need equations for calculating the overlap and gap connections. And in order to keep them scaled to the ORF distances, I use the same P not stop exponentiated by the number of bases, except here I make them positive so that they're always positive distances. They're always kind of like unfavorable. You have to go that distance to travel it. So the analogy I like to make is that of chutes and ladders, where chutes, ORFs, green, are free movements, negative distance, while ladders, gaps and overlaps, are a penalty to climb, positive distances. So to these equations, I make a few minor adjustments to the ORF scores depending on the start codon, the ribosomal binding site motif, and the GC frame plot. So the most common start codon is ATG, followed by GTG, and the least frequent is TTG. There are also a few others like CTG and some really extremely rare ones, but I don't really deal with those because not a lot is known about alternate start codons. So these 85, 10, and 5% changes also based on GC content. The high GC content phages, which have a lot of G's and C's, will favor GTG, bumping it up to 1/3 to as much as 40%. So the ribosomal binding site motif is based on the Scheindel-Garna motif of AGAG, which is essentially AGG, AGG, and has 27 different similar patterns. And I'm not going to really go over that, and you won't be able to do that yourself. So you have to use tools to look for that because it's really complex. But it's just good to know that it exists. So the GC frame plot is a strange feature of phage genomes, and so I'm gonna have to kind of discuss that. And it's gonna get complex, but hopefully at the end you'll be able to understand it, and if not, at least follow just the visual pattern. And to do this, I want to discuss the high GC content phages. So here's one such genome. I think this is mycophage L5, D5. And so these obviously have significantly longer non-coding ORFs due to stop codons occurring less randomly in the DNA, which you can clearly see here. The real complication is that they favor the third position for their max GC frame in coding genes. This means that if you look at the codons of a coding gene, there are far more G's and C's in the third position than in the first or second. So to explain this property, shown here is a tiny 5 codon gene where there are 2 GCs in all of the first positions. That's all the red positions. There's 1 GC in the second positions, all the blue, and then 4 G's and C's in the third positions, which is the black, but it looks like so this here should be black, but so you count up the G's and C's in each one of the positions. So 2, 1, 4. This means that the third position, the fourth, the number 4, is the max GC frame. So the reason for this bias is because regardless of GC content, genes still need to use the same universal amino acid frequency shared by all phages and life in general in order to perform the same functions. So to use the codons of this universal frequency, they accumulate the G's and C's in the degenerate third frame. For example, if you're a phage with lots of G's and C's in your genome, say 66%, and you need to code for serine, which is up here, the first base must be a T, and the second a C. For the third, you can choose any of the 4. You choose a G or C in order to use up G's and C's and save them, and save A's and T's for use in first and second positions, which generally do not allow the alternate nucleotide choices. The consequence of always having a G or C in the third position is that in the mirror reverse frame, so that's going to be if you're in the first frame, the -1 would be the mirror reverse. If you're in the second frame, the -2 would be in the mirror reverse. You always have a G or C in the first position. The first base of the 3 stop codons is a T. So you see far fewer stop codons than expected in the mirror reverse frame that shadows a coding ORF, and extremely long non-coding spurious ORFs occur, which really confuse gene finding methods. So here's one extremely long spurious ORF on the bottom in the -2 frame. That's the shadow of a +2 coding gene in that second frame. You can see many more that shadow the other coding genes as well. So to show how to use the GC frame plot for gene prediction, I've taken the tiny 5 codon gene and repeated it twice so that I have 2 genes, one after the other. So for the GC frame plot, rather than counting the G's and C's for each codon within a gene, you count the G's and C's of a window centered at that codon. Generally for Artemis and my programs, I use 120, but for my example, I'm only going to use 3. So here's a 3 codon window centered at the ATG. There's 1 GC in the first position, the red one. There are 0 in the second positions, the blue. And there are 2 in the third position, so 1, 0, 2. So the max GC frame at that codon position is the third position. Then moving to the next codon, still looking at a window of 3, we see 1 GC in the first positions, 0 in the second, and 3 in the third. So 1, 0, 3. And I move likewise through the rest of the codons, counting the G's and C's at each position, until finally at the end, I'm left with a bunch of sets of 3 positional scores. If I plot these 3 position sets as 3 lines, so the first position is a red line, the second is a blue line, and the third is a black line, you can see that the max GC frame from codon 1 to codon 5, that's going to be here, matches the gene occurrence of that first gene. And that where the max GC frame is blue, matches that second gene layout. So this is kind of probably confusing, but let me show you some real data. So this will show you how clear the signal really is. So here's the same high GC content genome that has those spurious non-coding ORFs. And you can see that the GC frame plot up at the top is created by the Artemis plot, which I'll show you how to use. And you should be able to clearly see the very distinct line patterns that match the green coding genes. So when you have the red line on top, you have coding genes in the +1 frame. When you have blue lines on top, you have coding genes in the second frame. When you have black on the top, you have coding genes in the third frame. And it's not just which is the top line, it's also the order. It's also, so when you have red, blue, black, it's in the first frame. When you have blue, black, red, you have second frame. When you have red, blue, black, you're in the again in the first frame. When you have black, red, blue, you're down in the third frame. And you can see this pattern repeated over and over again through the genome. And this helps you to pick out which are the coding genes because they follow that same pattern. So you can see that down here is, so they also, because the spurious non-coding ORFs, shadow ORFs also shadow the coding ORFs, it can kind of confuse you, but the order kind of tells you which one to take. And also you can see that down here, there's a non-coding ORF, and you can see that it doesn't really match, the GC frame plot doesn't match that one, so you know you're up on top. So hopefully this will become a little bit clearer when I manually answer the genome with Artemis, which I'm going to do next. So I think here. So, but before that, I want to talk a little bit about file types. So I'm going to be annotating the phage genome PHAX174, since it's a tiny genome, it only has 10 genes, and should be pretty accurate since it's a well-known genome and highly researched. I'm going to be starting with a FASTA file, which is the kind of the go-to file that contains sequence. And my idea is to get a features file, which will contain the genes themselves. And ideally, I want them blended together in a GenBank file, which is a middle. So this is kind of the basic layout of a FASTA file. So the line before any sequence starts with the greater than symbol, and then the next word after it before any space is a sequence ID, and then anything after the space is a comment. So anything after the greater than is a sequence itself. And a FASTA file can be nucleotides or amino acids, and usually you designate them by FNA or FAA to kind of distinguish, but you don't have to. You can just have the FASTA as a generic. GenBank files are pretty complex. There's very specific and strict terms like locus, features, origin. So you're going to want to have, you're going to want to let programs create that for you, which Artemis and my program will. All right, so I'm going to bring up, let's see, I'm going to bring up Artemis real quick, if I can. Oh, Artemis. No, it's not going to let me. File, open. Here we go. There we go. All right, so here's Artemis, and
Katelyn, I think you got your screen. Screen is not, we can't see your screen.
Is it not sharing? Okay, let me try to bring it up again. Yeah. Oh, here it is. Okay, so where is that? Yeah, I've apparently lost the screen.
No rush. Just let me know if you need anything. You
Yeah.
share and it's not a window anymore that you can pick. Oh, there we go.
Is it showing the whole screen or just the window itself?
You see the window like phax174.fn.
Yeah, so it's not showing the desktop. Let's see if it, so I'll open it. That's how you open it. Then
I guess now you have to open that. Yeah, switch to that window. Super annoying with Zoom, I think. I don't.
Yeah.
Entire desktop.
All right, so here I'll switch to the window. So hopefully it.
Yeah, I can see a sequence.
All right, so this is a Phax genome loaded in Artemis, and I've made this file available during the bit.ly. I don't know if that ended up in the Slack thing. I kind of gave you the link, but so this, as you can see on the top, it gives you the 6 different frames. And on the bottom down here, it gives you the DNA and the amino acid translations. You can see the various stop codons, and it tells you where you are in the genome. And if you're me, you can really see where the genes occurred just by looking at where the stop codons are. Because I've been looking at genomes for so long now that I just automatically see where things are. But one thing to help you, you go up here and you do the graph, the GC frame plot, and that shows you here. And I'm gonna actually change the window size from 120 up to like 200 just to show it more visible. And what this tells you is that you can kind of see, especially here, you know there's a gene because there's just this really long region, and it's very improbable that there's not a gene there. And so what you do is you kind of do create and you do new feature. Oh, no, I want to do new feature from base range. And so you do create feature from base range, and it creates a gene for you. And what you want to do is go back to the end, and I hit the stop codon perfectly, but the start codon I missed. And as you recall, there's ATG is the most common. There's also TTG and GTG. And you can just kind of drag this to probably the leftmost start codon. That's usually what phages are.
We can't see your changes if you're doing anything. Just want to let you know.
Oh, okay. Then yeah. Yeah. Let me see if I can share. All right, I'm just gonna load the, let me load the genome because it's not showing real time, I guess.
So in that case, Katelyn, probably you can tell more about, as you had discussed earlier, about these, then how to do, if that's not possible at this time.
I mean, I can try showing, I mean, I didn't really plan on, I should kind of planned on walking through the couple steps to how to do it, but I guess I have this, so I can try this again. So let me bring this up again. Where is Zoom? Share screen. Here. So is it at least showing the PowerPoint again?
Yeah.
Okay. So this was what Artemis looks like once you load it. This one already has the genes annotated because it's just a placeholder that I put the final genome. I didn't plan on having the genes. And so in this top screen, you can see the GC frame plot that I mentioned. And in all the bottom, you can see where the genes are, and you can see that the genes occur in these long open reading frames. and they really match the GC frame plot. So you have blue, black, red is up on this top. You have red, blue, black, which means down here. And then I'm going to skip this one just for now. And then when you have, like, when you're in the top frame again, it's that blue, black, red, blue, black, red. And when you're in the bottom again, it's that red, black, red, blue, black. The PHAX genome is interesting because not only does it have overlapping genes, but it has genes that are completely overlapped. So you can see that there's this gene A, and there's a smaller gene completely overlapped inside of it called B. And you can see in the GC frame plot, it really shows that in the second frame, you have this little peak here that shows you have another gene there. And so no current genome annotation program will actually find that for you. Like PHANOTATE won't, certainly Glimmer, Prodigal, anything else, GeneMark won't. I'm working on making that available for PHANOTATE, but it's really troublesome because there's just so much potential for adding false positives to the data. So a lot of this is that you have to use automated program to annotate a genome, and then you have to go back in and fill it out yourself. And this is something I've always been doing, and I kind of just realized that I have to do myself. And I've actually found other labs that actually have the same kind of procedure, that they have an automated steps that they annotated, and they have their students go through and actually manually curate the genes by adding in any missing genes or removing any false positives. And so with the PHAX genome was interesting. So out of the 3 programs, that of course, nothing would find this one because it completely overlaps, and nothing would find that because it's completely overlapped. And the same with this gene here. Those were all missing from PHANOTATE, as well as GeneMark, Glimmer, and Prodigal. And GeneMark and Glimmer both miss this tiny little one because they kind of miss a lot of small genes. And Prodigal had a false positive somewhere, I think, up here. And so you really have to open your automated annotation and go back in and fix it yourself by looking at the GC frame plot and looking for empty spaces. Do you see missing a gap where no gene occurs, that there potentially could be a gene, and you add it in yourself? And so I don't really have all that much to talk about because I had lessons that aren't apparently going to work. So I think I might have some stuff. Let's see, why are you not? Let me see. I think I have a little bit on how to run PHANOTATE, but not much. Is it still sharing?
Yeah.
Okay, so yeah, so here is that. So for automated gene annotation, rather than going in Artemis and actually doing it yourself, picking out every gene, you run a program that'll find them for you. And so I wrote PHANOTATE, and it's available as open source software, and you can get it on GitHub, or you can download it yourself using the Python pip package manager, which is really just single command. And so I've made it as easy and simple as possible for someone to run. The only thing is that to do it involves the dread command line, which most biologists just refuse to even use. But at this point, it's something you're just going to have to kind of learn to do, because it's really becoming kind of part of every operating system and procedure now. And so it used to be Linux only, but now it's kind of an integral part of Macintosh, and Windows even has the ability to use it with the Ubuntu installation package. And so PHANOTATE runs on the command line like all the other programs out there. And so to run terminal, if you're on a Mac, it's hidden in your applications, and it's down here called terminal. And it's really easy. You bring it up, and this is for Windows. You have to do, the Windows is more involved. It's a couple more steps. You install Ubuntu, and then you do the Windows, and it kind of installs it for you. You'll have to look at like an online how-to do it for Windows. I've done it on my other Windows machine. So once you have a terminal, it really is only 2 commands to download and install PHANOTATE. So here I show a couple more because I show how to download the PHAX genome that I put online as an example. So wget, which means webget, so bit.ly, the phax genome. And because bit.ly doesn't give it the right file name, I just rename it from phax174fna to .fna. And to install PHANOTATE, all it is a pip3 install PHANOTATE, and then you do -- user. And this is a common command to install any kind of Python package. And it's a really simple and easy method. And everything should kind of just install automatically, and it'll give you a whole bunch of output and just it'll run. And then to actually run PHANOTATE, it's a single command, phanotate.py. You give it the name of your genome, phax174.fna, and then you give it a -f for the what you want your output format to be. So I want the output format to be GenBank. And then I give it -o. I want the name of the output file, so phax.genbank.gb. And it runs. It should take a couple seconds. And when you're done, I do ls, which means list. It shows that in addition to the phax174.fna, I also have the .gb file. And I've already shown this because I didn't expect to, but essentially the output from PHANOTATE is identical to all the steps that would have been done during manual curation, except that it would have been missing these couple completely overlapping files, which I was going to show how to pick those out and add them, but since Artemis isn't working, I'll just say here that essentially you would have had this gene, this gene, this gene, this gene, this gene, this gene, and this. And you would have had to go in and identify something suspicious here because you see the GC frame plot and you add that gene. And then to second verify it, you would then take that sequence of the gene and potentially blast it or use some kind of functional analysis to figure out whether or not it's a true gene. And Artemis has that built in. You just right click on it and then say blast and it'll blast it for you. But I can't show that now. And Artemis has really great tools for essentially what it'll do is it'll find all your open reading frames and trim them down to the first start codon until you have all the potential true coding and false positives. And then you go through and you pick out which ones are wrong, which ones are right, but I can't show that here. Once you get, yep.
If you do want to troubleshoot that a little so you can get it working, it's totally fine if you want to give that a try.
I was looking at it, it was just it's not showing up. I don't know why.
Zoom isn't sharing an application.
Yeah.
Okay, just wanted to give you the option.
Yeah. I ran through yesterday and it worked, but not today. So
yeah. All good.
Yeah. So one of the things that PHANOTATE doesn't do is it doesn't give you functional annotation, and that's also part of that's one of the reasons people like pipelines. And so if you really refuse to run PHANOTATE on the command line, it's available on the Patrick website. So that's going to be full automation using Patrick. And it's really simple. You just point and click, upload something, and it gives you everything automatically. Let's see. Oh yeah, so the full automation using Patrick. And this is a website, the URL for it, and you can register. And it's a really simple, there's tutorials on how to do it. You can see the upload link down here. You just give it that FASTA file, the file FNA, and it'll give you your GenBank file with functional annotation. So it's really cool. But again, it won't really give you your complete 100% correct genome. So you'll still want to download it and use Artemis or some other visual genome browser to go through and either pick out any false positives or add in any false negatives. So if you're computer savvy and you also want to use Patrick to get the functional stuff, you can use, they do have command line interfaces. And so my boss, Rob Edwards, has a tutorial at this web address on how to submit a genome to the Patrick pipeline using the command line. And this allows you to submit hundreds or thousands of genomes in an automated fashion, rather than having to point and click and upload every individual file. And I've used this to upload, I think, 10,000 phage genomes. And I was going to walk through how to use the Patrick, but it's apparently not going to do interactive stuff. So I guess I will kind of conclude here on with lacking a lot of my stuff, but there was one thing that I want to mention a little bit before ending. And it's a problem that I've had a lot of students and other researchers encounter kind of unintentionally. And that is that you have to be aware that phage genomes are circular. And so what happens is that when you sequence a genome and then assemble it, the beginning and end in the sequenced file can be really arbitrarily anywhere along the genome, potentially even in the middle of a gene. And so what happens is that two genomes that are identical can actually look not identical. And so this is phage 1 and 2, or this is the same phage that I've just kind of respliced differently. And I've split phage 2 on one of the green orbs that are in the reverse frame. And I've had a lot of students that come to me and they'd say, hey, here's a bunch of different genes. Can you tell me a little bit about them? And I've come to find that they're actually all the same genome, which is quite common when you're doing sequencing and growth and stuff like that. And even with the PHAX genome, when I first downloaded it, it was actually split on a gene. And so this was the before. So this is the one that I've presented where you can see that I've put the origin of replication at the beginning, and then all the genes are nice full genes all the way along the length. And so these two that I've highlighted here, in the original annotation, they're split. And there's half at the beginning and half at the end. And the Artemis, kind of to show this join, uses this little red line that kind of tells that they're connected. And this makes a lot of analysis and just visual inspection and stuff like that really difficult and hard to look at too. And so you want to be aware that you might want to recircularize your genome before or after analysis, just to make sure you're not splitting genes, you're not missing genes like that. And so a common way to do this is to recircularize a genome on a common feature. And for my case, I recircularized on the origin of replication that was in their GenBank file, and I put it at the beginning. But you might also want to recircularize on like a certain gene, like an integrase or a terminase or a capsid, because this will allow you to also orient a bunch of genomes all together, which is what I had one student do, is that she had a bunch of Chromobacter phages, and they were all circular genomes, and she wanted to kind of align them all together so you could look at them and create a visual plot and look at the similarities between them. And so I actually wrote another little tool. It was called Circu-Align, and it will take as input a bunch of FASTA files or a single FASTA file, circularize them, and then find a common feature, and then align them together on that common feature. And this is what I use to recircularize and align the PHAX genome so that the origin of replication is at the front. And this one's really easy to use. There's instructions on how to do it. And I've had a couple students use it and really like it. So this is something to be aware of, that you need to potentially know that your genomes are circular, and for better annotations, you want to recircularize them so whole genes are not split. And I guess I'm going to leave off there for questions.
Awesome.
Any comments?
Thank you, Katelyn.
Yeah.
Super awesome to see all that. And we have a bunch of questions. So I'll start off reading some of them that we have, and then we'll see if we get more or if people want to chime in verbally after that. But we're going to read the ones that have been added first. So what is the codon window used to plot GC-plot?
So that would be the codon window. It's the GC frame plot, and I'll go back to that. And it's really used so it's used to score ORFs, potentially. So you have coding ORFs, and you need to figure out whether they're coding or not. And so you use it to kind of scale the ORFs, whether or not they match the general consensus of the background genome. And it's used in this case, because I don't want to go into how to do that analytically, but it's really useful when just visually inspecting the genome in Artemis or any other visual browser to look at the GC frame plot so that you can kind of see which genes match the GC frame plot. It gives you kind of a roadmap on how to determine which is the correct coding ORF and which isn't.
Okay, so many phages also encode for their own tRNA. Can we annotate the same using PHANOTATE? How about UTR present within the phage genome?
Yep, and actually that's a great question. And so one of the first problems that I ran into while developing PHANOTATE was that not all genes are protein coding genes. So tRNA is a gene that encodes an RNA. And so if I don't mask out the non-protein coding genes, there's a lot of false positives that get added to the predictions. And so I added the ability to look for tRNAs and add them to the program. And so I use tRNA scan. And so you have to install that yourself. And then if it's there, PHANOTATE will run it and then find the tRNAs and add them to the GenBank file for you. And there's a whole bunch of other different types of genes and features that I want to add to PHANOTATE. Things like miscellaneous RNA and temp RNA and non-coding ncRNA. And there's other things like origin of replication and ATT at sites that are all kind of functional things, but that aren't protein encoding genes. And so the more of those I can find and add to PHANOTATE, the better it is at predicting the path of the genes.
Got it. Okay, so dear Katelyn, just need a little confirmation. Does it mean when we're using PHANOTATE, should we recheck the result using Artemis to make sure we don't miss such a gene?
Yep, you always do that. And so that was something where like none of the programs themselves are 100% correct. And certainly PHANOTATE isn't. It's one of the more accurate ones, but there's always, it's not 100% yet, 100% correct yet, although I'm trying to get it there. And so you always want to post automated annotation. So after running the automated, go through and visually inspect the genome yourself and figure out whether or not it looks right. And it's oddly enough, yeah, like I said, it's something that I had just kind of learned to do myself, and that when talking to other labs, they've kind of come to the same conclusion. I think one of the other labs, the one that does the mycophage stuff, the SEA phages, they have the same kind of protocol where they use, I think rather than Artemis, they use DNA master to get a GenBank, and then they go back in and do the manual curation to make sure that the DNA master stuff's correct. And it's just both of our labs came to the same conclusion that you have to automate it, and then there's always a step of manually curating things.
Yeah, last time our speaker was presenting DNA Master. She was from Hatfu's lab, Debbie, if you might know.
Yeah, it was really funny because he was here visiting and we were talking about it. We were both it was interesting that we both realized that there's always this need for manual curation, that computers just aren't smart enough yet.
Good, we're all still needed. Okay, here's one from Piotr. Thanks for the interesting talk, Katelyn. I'm very willing to try PHANOTATE as a glimmer replacement in my pipeline. What is your opinion about language model usage for gene prediction? Unsupervised deep learning approach trained on a set of brackets perfect reference genes would sound as the next step in that research area.
So one of the things that I've always shied away from in any kind of computational machine learning program that I develop is references, because it's just like, it's the lowest hanging fruit is using a reference database to predict stuff, because it's really easy. And that's why things like GeneMark and Glimmer work so well, is that they use a reference database. And so when you take your PHY genome and if you do a single mutation to it and then you want to annotate again, all the other programs use a reference and they already know what the genome looks like. And so using references it works really, really well for things that are very similar to things you already know. And that's the one benefit of it. But anytime you get something that's highly divergent or just very different, which is quite common of phages, using reference databases can lead to just missing a lot of stuff. And so that's one of the reasons I kind of get away from reference-based learning anytime I deal with phages. And I've thought about actually adding reference-based learning to PHANOTATE because it's just really easy. I mean, it would make some prediction that much quicker. It would predict the things that we already know very easily and stuff like that. But yeah, using Glimmer and models, I mean, that's what Glimmer does, is that it looks, well, the Glimmer can run two ways. So Glimmer will run by itself using the pre-computed models, but it also has like the G3 from scratch script, which can create your gene model based on the input genome itself. And that's where it kind of really goes kind of more awry, because it uses a long arse program, which doesn't really give you kind of the best result, and especially for phages. And so I guess using amino acid coding and reference models and deep learning, I guess neural networks can work, but with phages, it's really problematic because phages are so diverse that it's the phage dark matter is what it's called, is that 70% of any phage genome and 70% of the genes of a phage are completely unknown. And so you'll run into that a lot with phages using reference data.
Got it, yeah. I just want to pause here because we're at the 1 hour mark, but are we going to the 1.5 hour mark today, Katelyn, or what did you guys want?
We have I think we have a couple more questions. One is—
Yeah, only if you need to.
Okay.
Daniel is asking questions.
Stay on for a few more for the end of the questions then. Great.
I have 2 more questions, I think.
Okay. Okay, from Dan Nelson, thanks for the talk. I manually sequenced a phage genome in 2003 by primer walking and Sanger sequencing, and annotated it by hand using ORF Finder and GeneMark. There are hundreds/thousands of phage sequenced during this time frame before next-gen sequencing and bioinformatic workflows. If one were to use your approach to re-annotate these older genomes, do you expect new genes would be found or significant changes?
Definitely, yeah, I definitely think new genes would be found, especially with my program versus the old ones, because a lot of the old ones erred on the side of caution and really made sure to have only very strong positive genes. And so they kind of excluded a lot of lower weakly scoring genes. And also the fact that all the gene finding programs, PHANOTATE included, use an arbitrary cutoff of 90 base pairs, so that anything smaller than that is just completely ignored, so I'm sure we're ignoring a very large percentage of genes out there that are sub 30 amino acids. There's a whole category from that we're kind of skipping. And so using the newer programs, GeneMark and Glimmer shouldn't change. I don't think Glimmer has been updated in the last 10 years. 3.02 was the last update 10 years ago. Prodigal hasn't been really updated in 10 years, although it's mostly reference free, so it doesn't really need to be. And GeneMark's upgraded quite often, but I'm not sure about the databases, so I don't think too much would change if you reran GeneMark or Glimmer, but you'd definitely get different results with PHANOTATE, I believe. You'd probably get a little bit more of the lower weakly scoring things that are potentially protein encoding genes. It's just harder to verify whether they actually are, because you need metabolomic data, and that's very hard to come by. And it really takes manual curation of you'd have to go through and look at which genes and say whether or not any new genes added are right or not.
Here's one. What advantages are there of PHANOTATE over Artemis?
So one of the things is that PHANOTATE will do it for you. So if you're only annotating a small genome, like I did with PHYX with 10 genes, you can do it really simply. Like I would have, if it would have worked, it takes just a couple minutes to actually pick out the 10 genes. But if you have 100 genes, it can be very time consuming to do just one phage. And if you have 100 phages or 1,000 phages, it just becomes impossible to actually manually curate a genome. And so that's where PHANOTATE comes into its own, where you want to run PHANOTATE to automatically annotate a large set of phage genomes. And potentially, if you can, maybe go through and review some of them to see how well they're doing. It's just Artemis is very good to manually curate yourself, but it's just you can't do it for large genomes or a large number of genomes.
Okay, from Marcin Lubaki, does GC frame plot differ between different gene classes?
I don't believe it changes due to different classes. I'm not sure, I haven't done any analysis on that. It does change based on the GC content. So the high GC content phages will favor the third frame usually for the reasons I mentioned, is it because you want to store your G's and C's in third frame. If you have, for the low GC content genomes, it's the opposite, that they pack up their T's and A's in that last third frame. So the highest GCs in the first or second frame. And so that's the only way it changes, but I don't believe it changes across gene classes. Although it could, because certain gene classes have different amino acid frequencies, but I'm not really sure how much it changes.
Okay, thank you. Okay, that's the last question I see. We are going to make resources available, right, Ermi? We have from Katelyn, we have kind of some links, and we have a Slack channel on the Phage directory Slack that I'm going to add into the chat, or maybe if Jan is listening, he can add it. We have a Phage Bioinformatics channel within that. And that's where we're kind of directing anyone who's coming to these webinars, who wants to get more help, help each other and find like, oh, I missed this, link or which tool. And Katelyn, you're welcome to join in and chime in on any questions that come up in there too.
Yeah, if you can send me the link to the Slack, I can join them.
Great.
Yeah, that would be great.
I will make sure you get.
I can give all the.
Yeah, and there were some questions on your facts also about prediction on temperate versus lighting pages, but probably we can't cover them today. But would you like to just tell a little bit about the experimental validation, if there have been any studies, because there were several questions on the prediction of phages as temperate by facts, whereas the experimental studies show them to be lighting. So how much of this can we expect between the prediction and actual experimental validation?
Yeah, so the experimental and the validation was actually there was a cool feature of the facts that actually that I noticed is that when I developed facts, it was 10 years ago almost at this point. And so at the time we didn't have very many phage genomes. There was only about 300-ish that we actually knew that were experimentally validated. And that's what I had to use. And when I ran the accuracy, whether or not how correct PHANOTATE was, one of the things that happened was that there was the one genome that it actually was wrong on was, I think, a mycophage D5, which was actually part of, I think, the SEA phages, which was interesting. He was the same person that did that. And it was wrong because the mycophage itself, it was predicted as temperate, except it had been shown to never be temperate. And what happened is that it has an integrase, it has a functional integrase, I think, but it just was never proven to ever integrate. And so that was the one that it got wrong. And other than that, I haven't really done too many experimental validation on it because at the time there just wasn't that much data. And I haven't really updated the database only because the funding, I've been busy with other stuff like that.
Okay.
So there's not a whole lot of data.
Right. One more question. Do you have a video tutorial to use facts? I mean, someone wants to use facts. So would you recommend any video tutorial for that?
I don't think I have a video tutorial, but I've made facts, because the facts runs on the command line, but I've also made an online thing, which should be pretty easy. You can just upload. I mean, when you go to the page and you do upload, it gives you a box, and it should be really simple to upload. It's 2 clicks, I believe. You choose your FAA file, and then you click submit, and then it gives you a prediction. And so one of the things is if you don't have an FAA file, PHANOTATE will actually give you one. So if you have just your FNA file, the DNA backbone, you can use PHANOTATE to get the FAA file using that -F command. So -F, so PHANOTATE.py -FAAA, and then your FNA file. So I can give some of these commands on the Slack or something potentially.
Okay. So with that, shall we close?
Yep.
So thank you so much, Kate, for coming. And very not often we get to see how applications run actually behind the scene cases. We normally use feed our sequences and get the output files, but this was a very new learning for most of us. So thank you so much, and glad that you're joining the Slack channels because there'll be several questions posted there as well. And if you could take those, I was particularly interested to know that how amino acid composition, I mean, how your gene prediction in annotation, you factor in the amino acid composition of the sequence. I don't think you could have covered it, but maybe we could take this discussion in Slack.
Yeah, sure. Yeah, hopefully I'll be able to cover some of the stuff that I would have been able to show in the interactive session that didn't work. So
right.
The Slack will be there. I'll be there for the Slack.
Wonderful. Okay, then thank you very much, all the participants, and thank you so much. It's 9 o'clock in the morning for you, I think, and you're done with a great talk already. The great start of the day for you, and we have a late evening time in India and Asia. So I thank and Jessica, thank you so much. Jessica has been keeping very busy this off late, so I think that's it from us, and let's hope that we'll have a next webinar anytime soon.
Yep, great. Yeah, thanks everyone for coming. We'll be writing up a recap of this eventually in Capsid and Tail and posting the recording on our YouTube channel. So that's all good with you, Katelyn. So yeah.
Cool. Thank you.
All right. Thank you so much. Bye-bye, everybody. Bye. Good evening and good day.